Hi, Kiara.
Hi.
That's how we got. I'm also coming at you from my office this morning because I have some lab work to do this afternoon. So hopefully the connection is strong enough in here. I tested it out yesterday and it seemed to work fine. But with technology, I swear, I feel like you just never know. So hopefully this works out good. If for whatever reason we get cut off or it becomes, like the connection seems poor, I will record anything that I want to get to immediately and post it on Blackboard. But hopefully, like I said, I tested it yesterday. Hopefully it'll be okay. For those of you just joining us, please make sure you open up your browser and open our Blackboard course site and click on week 2. There's going to be some documents there that I'm going to review with you guys and 2 papers that we're going to take a look at today, just to get you practicing reading primary literature. Hi, Dr. Parvisi. Oh, go. I think we almost got it, 36. So we're waiting on, I think there's 39 in this class. Oh, but Professor, you're here, so that's not what I was thinking. Let me see.
Morning.
Morning. All right. All right, we're going to go ahead and get started. I am going to start right on the dot, assuming I don't get disrupted by children. But now that my kids are back in daycare, that shouldn't be an issue moving forward, unless of course they close, but let's really hope that doesn't happen. All right, guys. So I know that we started our course last week, but I didn't really formally get a chance to introduce myself to you personally. I'm Professor Ellis. Many, if not all of you, have had me already at some point because you are all bio majors, and I teach a lot of the required courses for bio majors. So you probably all had me in biochem, genetics, let's see, molecular biology. I taught intro bio, so some of you might have had me 4 times up until this point. Alexa, you took my lab course last semester, so that's a new one that I teach. So yeah, I know most of you, if not all of you, fairly well. So I just wanted to say welcome. And today we're going to be focused on working through the process of reading primary literature. We're going to talk a little bit more about the project that's upcoming and answer any questions that you guys might have about the project. Professor Parvisi, do you want to introduce yourself since you're here?
Sure. Yes. Hi there. So I am Professor Parvisi, and because so many of you enrolled in this course, Professor Ellis decided that some help would be much needed, and I'm more than happy to be here to basically help her with listening to and grading about a half of you and your projects. So once things become clear as to what you're doing and you're sort of on the ball, starting to roll with your projects, I am someone who you can reach out to. Just a little bit about me, I'm a medical school graduate, and for the most part, I've done a lot of anatomy and physiology teaching. So, in addition to this course, if you ever have any questions about, what it's like to be a doctor or healthcare professional or, something like that, I'm also happy to answer questions, but I'm primarily here to help Professor Ellis with what she's doing. Okay, so nice to meet you all.
Yeah, so typically this course is limited to about 30 students. That's actually the high end. I really like it to be around 20 or 25, because each of you are going to be required to deliver your own oral presentation about your paper, which takes some time. And then you're all also doing a poster presentation. You're working in groups on some other projects towards the end of the semester. You're working individually on others. And so it's all project-based, there's no exams. So it takes a little bit longer for me to grade things like projects and presentations versus exams, which is just sort of kind of going through the pile, right? And so in order to make this class more efficient and fair for you guys, so that you have more attention from your professors, When it comes time to do oral presentations and poster presentations, we will split up into different groups. And some of you will be presenting to me and also your peers, and then some of you will be presenting to Professor Parvizi and then your peers in that one too. And so it's about 40 in here, so we'll probably split it 20 and 20. And so let's talk a little bit about and answer some questions about your upcoming project, which is the oral presentation. Oops, no, oh, I do have to talk about that too. So let me get, and Professor Parvizi, I'm gonna make you a co-host, just in case somebody enters the waiting room and I don't see it. Would you mind admitting them to the course for me?
Sure. Yeah.
Thank you. And also, if you wouldn't mind taking note of who's present, because attendance is required in this course. So just like quickly jot down who's here, and that will be, and then we'll touch base later to talk about that. Okay.
Sounds good.
All right. So today's agenda will be focused on tips and tricks you can use to analyze primary literature. And so reading scientific literature is not at all the same process or the same experience as reading, magazine articles or blogs or things you might find on the internet, news stories or books, textbooks and things. It's totally different kind of reading, and it takes a little bit of a different approach. So I'm going to talk a little bit more about that to make sure that you can get through your papers that you're going to be choosing and understanding all the details. So we're going to be focused really on that. And then we're going to talk a little bit, we're going to do a little bit of a discussion about exercise 4, which was presented to you in the first Top Hat chapter. So just a reminder to you, by the end of this week, so on Friday, January 29th, you are required to submit a PDF of your chosen paper via Blackboard. So there will be a Blackboard link under week 2. It'll also be found under assignments, so it'll be in different, it's all the same link, but I like to try to, put links to these assignments in lots of places so that you don't miss them. And so you'll be able to find a place where you have to upload your PDF. Some of you might have already submitted a PDF to me, but if you wouldn't mind also submitting your PDF copy of the paper to this link online. Okay, so what are you doing with that paper? So last week I asked you to please choose a topic of interest, something in science that's always intrigued you. You're all bio majors, you've all had several courses of different biology courses, some health psychology courses maybe, if you are have a pre-ben minor or something like that, or you intend to go into the health profession. You guys have some natural curiosities that you might want to explore a little deeper. And that's where this course serves 2 purposes. One, it helps you to communicate scientific information to other scientists and gets you familiar with how we communicate with each other. But it also helps you to read technical information and try to repackage that in plain language for anybody to understand. And so that's what we call the public. People who have no expertise in science, you have to be getting good at talking to people like that. Especially now, it becomes even more important now. As we're all trying to ride out this pandemic, vaccines are becoming available, treatments, public health measures, and a lot of people are confused. And it's sort of up to us as scientists to be able to talk to people in a way that's approachable for people to understand as well. And so that's what this course is all about. But it also gives you another opportunity to dive deep into a topic that you're already very interested in. So for example, I'm getting lots of different topics like cancer genetics. I got some stuff about oral health, because that person's interested in dental school. CRISPR has come up a few times. The stuff that you guys are interested, somebody wants to know more about Ebola virus. The biology of cosmetic surgery. I think that's the first time I've ever seen something like that, which is pretty great. I'm excited to hear about that one. We've got childhood cancer treatments, breast cancer, rheumatoid arthritis, all kinds of topics all over the place. And so you get to dive real deep into a topic that you're already very interested in and try to practice communicating that topic and those ideas. Also, you get to learn about all different types of science in this class, because lots of you have ideas that don't necessarily overlap. And so, you know, for anybody who's always been kind of curious about something like a bioelectronic interface, like using your mind to control computer impulses and things, like maybe you never would have read that paper on your own, but you know, you kind of want to look into it a little deeper. And so here's your opportunity to attend a presentation where someone's going to be talking about that. All right, so hopefully you got me your topic. I'm still missing, looks like 2 people who haven't gotten me their topic. That's very critical that you pick a topic, because this week you've got to pick a paper about that topic. Many of you who emailed me last week said, I have this topic, but I don't really know how to, you know, find a good paper. And I sent you several links to look at of secondary resources that talk about different recent advances in that area of science. And so that will help you as a sort of a jumping off point for you to find a primary literature paper. And so I'm looking for you to pick a paper that has the structure of introduction, methods, results, conclusion. It has to have that structure. If your paper doesn't have those headers, you're probably reading a review paper or some other type of secondary resource. And so that's a good place to start, sort of getting an idea of what types of research are surrounding that topic, but you can't actually choose that paper. So I'm going to be looking at all the papers to make sure you're choosing papers from peer-reviewed journals, papers that follow the correct format. And then you got to start digging into that paper pretty deep so that you can write your first graded assignment, which is the paper summary. Do I talk about, so I don't think I have a slide about that right now, but so if you go to our course website under week 2, you're going to see some resources there. This is how you got to class, because that's where the Zoom link was. But you're also going to see these 2 documents, a review of paper summaries and how to read a scientific paper. These are 2 documents that I'm going to talk about in class today. But if you look at this one here, the review of paper summaries. Oh, I don't know why that's not just. Okay, fine. Oh, because it's not a PDF. I'll fix that for the future. Sorry. So here's the review of paper summaries. This is sort of, these are the questions you should be asking yourself as you read through your paper. And the answers to these questions are going to become your summary. You want to write your summary as a narrative. So make sure you don't break up your summary into different headers. You're telling me a story about your paper. So you're not going to give me introduction in a couple of sentences, methods in a couple of sentences. Write it as though it's a story. It doesn't need those headers, but you want to make sure you're answering these questions. What is the big problem? What is the big overarching issue that this paper tries to address? What is the specific question the experiments in this paper try to address? Summarize the background information. What was the approach? Is it a clinical study? Is it a field study? Are we doing collections? Are we doing more basic experimentation, sort of like I have a hypothesis and I'm going to try these different things? That's all under the approach. What are the major results? What are the figures and tables that support them? And so you make sure that you can answer each of these questions after you finish your summary. This will definitely help you create one. If you sort of just answer these questions first and then just sort of rewrite it as a narrative. So this will be really useful for you. And some of this is what we're going to explore today. How do I identify what the approach is? How do I identify what the limitations of the study are, and so on? So hopefully we'll, if I ever stop talking, we'll get to that and start going through some of those things. Okay. How to read a scientific paper is another document that's very similar to the paper summary, but it basically is a more formal guideline way for you to approach reading scientific literature. Many of you in this class have already seen primary literature. You've probably already had to read some of this for other research papers and other courses. But you might not realize there's actually a pretty systematic approach to how you can read these things, especially when you're first starting out. When I first started reading scientific papers, I kind of did this thing where, you know, I got my paper and I would read it from cover to cover and be like, okay. And then usually when I would put that paper down, I would say to myself, I have no idea what I just read. I read, my eyes moved, and I read words, but I have no idea what just happened. I can't really explain what this paper is talking about. And so cutting it down like this in sort of these different sections kind of simplifies the process. This is really technical language and it might help you a little bit. And so again, we're going to go through this process with a couple of excerpts from different papers that I've chosen for us today. But this, again, refer to this often as you're reading through your paper. Not while you're choosing. You might want to choose a paper first, like, explore around a little bit. But then once you've settled on a paper, that's when you pull this out and start to systematically digest the information within that paper. And so this one's also available to you. If you are having trouble opening the Word documents, let me know, and I'll upload stuff as PDFs. I'll try to do that anyway from now moving forward. I sort of rushed putting this stuff up online last night. I apologize. This is a link to the rubric for the paper summary. So you're going to see things like this in week 2. Anything we talk about in week 2, I'm going to put in one place so it's easy for you to find. But later you might not remember, like, where did she talk about the paper summary? Or I need some specifics about that assignment. You might not remember what week it's in. I've also created this tab called assignments, which is going to have all of your rubrics, everything that you need to complete the assignments, upload links and things like that. So if you need to find something, if you're like, I can't find that link to upload this thing, it's always going to be under assignments. All of the information will be found here. So this is the rubric for the paper summary. This is what I will be following as I make sure that you have all of the appropriate parts of your summary. Did you write a good summary? And if you did, you should be fulfilling all of these criteria. And of course, being able to answer those questions that I gave you in reviewing the paper summaries. So I will have this one. This is actually what you're going to get back from me as a grade. And then I'll also be using that reviewing the paper summaries document to make sure that you are getting all the points that you need in your summary. Any questions about what's expected of you for next week? So by the end of this week, you need to submit a PDF of your paper via Blackboard. It's going to be under that assignments link. So you should be able, you'll find an upload link there. And then you also got to start thinking about your summary, because your summaries are going to be due at the end of next week. So you got to pick a paper and then just start getting into it. Start getting into that paper. Any questions about that assignment so far?
Hi. In the document that you just had opened, it said something is due tomorrow.
Oh, really? Nothing is due tomorrow. Which document?
Okay. It was like the one about how to read the paper.
It said it's due on the 20. It's one. It was
Yeah,
at the
hold
top
that.
paragraph. The 1 due on
Oh
27.
yeah. Oh, I'll remove that.
Okay.
Sorry, that's just from last year. I didn't get a chance to remove some of those dates. Ooh, thank you for catching that. Yeah, I'll go and make sure that's removed and clean up some of that stuff. Actually, while we're on the subject of due dates, this is a document you guys are probably seeing on my screen right now that I'm in the process of creating for you. This is the weekly due date checklist. If any of you guys have me in the fall semester for online learning, this will look familiar to you. This is something I like to do for all of my online courses, which is, it's tough. You can't just ask me, you can't just come into my office and ask me what's going on, what's due. And so this checklist becomes more important because it gives you information about which exercises are due, how much they're worth, and where to find them. So for example, this week, I told you this already, but and I'm sorry that I didn't get this to you sooner. As we are working out how to do things like poster presentations online and oral presentations online, I have to kind of shift around some of the due dates for things like rough drafts to make sure I have enough time to get you feedback. And so this document is still in flux, but I'm hoping to get to finish it today. So for today, so this is Tuesday, you just have to attend class. Simply attending class gets you these 2 half points. I don't even think we're going to be able to discuss exercise 5 because that's normally something we do as a group, and I don't know how that's going to work for this format. But we can do this one. You'll have to submit a PDF of your paper on Blackboard for a point, and then all of your your chapter 1 top hat exercises are due by the end of this module. So that's going to be Monday, February 1st. So make sure, if you haven't already, enroll for Top Hat. I sent out invitations last week. A couple of you emailed me and said you didn't get them, so I sent them again. I can keep sending them over and over until you get it. So let me know if you still can't access this or you can't figure out how to sign up for this. But then you read through chapter 1. It won't take you too long. And then there's little exercises in there. Read this, watch this video, take notes and submit to Blackboard. There's stuff that you do within Top Hat. And so this document is meant, I'm creating this to help you figure out what's due every week and where to go to submit things and so on. So it's not super confusing as to where you're supposed to be and what you're supposed to be doing. So I'm almost done with this. I will get it to you very, very soon, I swear. But this is something I'm working on. Since we were talking about due dates, I thought I'd bring it up. Your top hat exercises. So just to remind you, so I'm opening it up now. All right, so this is one, the preface was this, you know, did you read the syllabus? Did you read this little intro? And then you just give me a thumbs up, and that's it. That's all you had to do for that, which was last week. I will be checking these each week to make sure that everybody is staying on task with their exercises. This is chapter 1. This is what we're focused on for the next couple of weeks, reading and interpreting primary literature. Next week, we're going to be doing chapter 2, which is presenting your research. And then we'll stick with chapter 2 for quite some time, quite a few weeks, because that's going to cover oral presentation of information and then poster presentations. But this is something I wrote myself based on, you know, scientific research, but also my drawing from my own experience. And so you're required to read through these and perform the exercises. So you'll see this is exercise 1. That's what I'm talking about. When I say exercise 1 from Top Hat is due, you need to come here, read this video, submit something to Blackboard, and then participate in these things. And it looks like some people have already started that. Great job, you guys. And so any questions about how to access Top Hat, what to be doing there, this is meant to serve as a textbook for you, an online text, but really, really specific for your needs, because I wrote this for this class. There might have been something in the chat. Let me just make sure. Someone is asking, so Katie, you ask if the paper we submit is different from the topic we originally chose, should we email you ahead of time? If you end up going down a rabbit hole in a different place, Katie, I don't know, hold on, I gotta see if I can see you here. If you end up just sort of in a different place and you really wanna do this, but you told me originally it was a different topic, me asking you to choose a topic was just me trying to get you motivated and get you started. If you pick a paper and you love it and it's a different topic, that's fine with me. Just tell me that when you send me the paper, when you upload the link, that you've chosen a different topic, so that later when I start to group you based on overlapping interests, I'm not grouping you based on an outdated topic. But yeah, you can change your topic if you find a paper that you really, really like. I am totally okay with that. I'd rather you choose a paper you really like than a paper you're kind of so-so about, because this paper is going to follow you for the whole semester. So I'd rather you pick something you really want to dig into and enjoy. So that's fine. Any other questions? So again, this week, you got to work on your chapter 1 Top Hat exercises, which are due by Monday. And you need to get me a PDF of your paper, your chosen paper, what you are absolutely going to be presenting on in a few weeks. Submit that link to Blackboard. All right. Okay, so, and then again, you can find the paper summary rubrics. All of that information should be found under week 2, how to read through scientific literature, how to write your summary. A lot of people ask, like, how many pages and things like that, like 2 to 3 pages. Just follow the guidelines that I've given you on the review of paper summaries and the rubric, and you should have plenty to talk about. Please don't make it more than 5 pages. That's not a summary. That's just rewriting the paper. So yeah, I usually, you know, I usually require something like 2 to 3 pages, maybe single spaced 2 to 3 pages. So not a double spaced 2 to 3 pages, that's like 2 paragraphs. So I usually read stuff in single space because that's what I'm used to reading with scientific papers and things like that. So single spaced 2 pages or so. I don't care what font you use. I don't care. I care about content. So I don't really care about length. If the content is there, it doesn't really matter to me how long or short it is. Okay, so let's talk about how you read primary literature. A lot of people, when they come to me asking for advice about how do I study for the MCAT, one of the sections they're the most worried about is the critical thinking and analysis section on the MCAT. And this a lot of that is reading something, interpreting that, using information you already know and applying it to this problem. And that isn't exercising critical thinking. And so when people ask me, how do I study for that part of the MCAT? I give them two answers. One, I say, hopefully you've been building that skill while you're in college, because it is not a skill that you can crash for. It's not something that you can just get good at overnight. It's a process. And the other thing I tell them is read primary literature, because that really does force you to improve your reading comprehension. Do I understand what I'm reading? And then reinterpret it in a way that you can understand and remember it. So trying to repackage it into a framework that already exists in your brain. Where can I kind of put this? And how do I use this information? All of this is critical thinking and problem solving. And that's really what that section of the MCAT is trying to test. That's really what you have to do if you decide to go to graduate school for research, or if you decide to go to industry as a job. You need to be able to read information and apply information to a new and evolving problem. So reading a scientific paper will help you practice that skill. It's not like reading in a science blog or reading things on Reddit. Usually you have to take notes when you're reading a scientific paper. I was doing this as a postdoc. Postdoc is the position you get after you graduated with a PhD. And I was still, every time I read a paper, I would have a sheet of paper next to me. And as I'm going through it, I'm writing out key points about each of those different things. The key point from the introduction, which tends to be the hypothesis. The key approach, is this a field study? Is this a clinical study? Or is this an experiment done in the lab? Is this a cell culture experiment? Is this a genetic experiment? Three of the major results, three of the major conclusions. I usually write notes about, if this is a major result, what figure in my paper supports that conclusion? And so it really, I was taking notes on primary literature, and to some extent, depending on the paper, sometimes I still do. If it's a paper I feel like I want to use that methodology in my own experiments, I still take notes on these papers. So you can't just simply read the paper and understand it. You really do have to take notes as you're working through it. You got to read it multiple times. There is not a single instance I can think of that when I was reading a paper to try and understand something about my research or something about a problem that I was facing, I never read it once. There's no way you can just read it once and understand it. You got to read it multiple times. The only time I ever read a scientific paper once is if I'm reading for fun. And so that's if I'm reading some kind of cutting edge, science that has nothing to do with my field. Maybe I'll read that once just because I'm curious, but that's about it. If I really want to use the literature, you got to read it multiple times. And then often I have to look up other sources to understand what I'm reading. And so, for example, if I'm reading a cholera paper and they talk about a vaccine that I hadn't heard of, I will go and look for details about that vaccine to make sure I understand the rest of the paper. So usually you have to do that as you work through the introduction of the paper. They might be talking about concepts that you're not totally familiar with. And so you might have to go to Wikipedia or something and look up a concept just so you understand it, and then return to your paper. Okay, now I know what immunological protection means, and so I can go back to my paper and continue reading. So that means reading one paper might take you a while, might take you some time. And so that's why I want you to submit your PDF of your paper on Friday so that you have the weekend to really start digging through and getting into that paper. Which leaves you, if you can do that over Saturday and Sunday, read it multiple times and take notes, that gives you the whole week to create that summary. So those of you might send me a panicked email on Friday night, like, I can't pick a paper. Like, you should send me that panicked email on Thursday night, not Friday, so that I can help you. You really should have a paper ready to go when you enter your weekend. So you can slowly at your own pace work through that, because it's going to take you some time. Okay, so the danger in taking notes on primary literature is that the temptation is to copy words or phrases directly from the literature into your notes. And I highly recommend you don't do that. Copying an author's exact words and putting them in your summary is a form of plagiarism. So when you go to take notes and then you're trying to write a summary based on the notes you took, you might be accidentally plagiarizing something you read the first time. And so when you submit your paper summaries, I'm going to be using Turnitin. And so I'm going to be looking for similarities, and I will allow you to see your own similarity report, because my goal in using Turnitin and using the similarity report is not to catch people cheating and to catch people plagiarizing, because often when you're summarizing primary literature for the first time, it's very difficult to summarize technical information, and it takes practice. And so I'm not looking to ding anybody on plagiarism. I mean, if it's 97% similar, okay, that's basically plagiarism. You just cut and paste right from the paper. But if it's something like 30%, which would be a red flag to any humanities professor, to me, that's telling me this person is having a hard time paraphrasing technical information and needs more practice. And so I'm going to allow you to see that. And so you can, you can upload your document. I'll let you do it multiple times. So I'll set up the settings so you can submit your paper multiple times, so that if you see like, oh, my similarity report is like 50%, I can't submit that. Go back to the drawing board and try again and resubmit and see how you do on that one. A lot of you, the best I've ever seen in this class was a 5% similarity report. That was amazing to me, that that person was able to repackage and paraphrase technical information to almost completely her own words. But it's rare when I see that. Usually I get something in the 20 to 30% range. And that's not something you could ever publish. You would get cited for plagiarism if you tried to publish something like that. And so citing it away, simply citing it, doesn't make it okay for you to write an author's exact words. You have to get good at paraphrasing, because eventually you will have to do this on your own. If you pursue research as a career, even in the medical field, you might be doing research as a fellowship in your fellowship years. You've got to get good at paraphrasing information, because when you go to write your own paper someday, you will have to use previous literature to back up, give context to your work and back up what you're saying. And in order to use previous studies, you're going to have to be able to rewrite that and paraphrase it in words that are not exactly the same as the original study, or you will not get published. You'll just get an angry email from the editor that said, this is blatant plagiarism. So this is going to give you some nice practice on how to do that, how to distinguish authors' words from your own. And part of that starts with taking notes. When you go to take notes, avoid the temptation of writing word for word what your paper is saying in your notes. Try to start that paraphrasing practice right during the note-taking process. Okay. So this is an example of what I'm talking about. Would this be considered plagiarism? Yes. So here's our original. Let's see if I'll make this bigger so you can actually see it. There we go. So you can actually read it. A very virulent isolate of Alternaria molly, the causative agent of apple blotch, was found to produce 2 major host-specific toxins and 5 minor ones in liquid culture. The minor toxins were less active than the major ones, but were still specifically toxic to the plants, which are susceptible to the pathogen. Blah, 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 blah. What on earth is this thing saying? And so how I would look at that, I'm saying, okay, so we've got some type of pathogen, which is a bacteria that causes illness. It specifically causes apple blotch. It makes toxins. Let's see, so 2 major host-specific topics. So it makes a lot of different toxins, some of which are active, some of which are not, some of which are specifically toxic to their plant host, some of which are not. And so this is sort of the notes that what I'm saying are the notes that I would be taking. So the first attempt at rewriting this is plagiarism. So they cited it, but that's not simply enough. You can't just cite this away. Komodo et al. Found that a very virulent isolate of Alternaria molly, the causative agent of apple blotch disease, this entire first half of this paragraph is almost exactly the same, sort of rearranged, but same words. That's plagiarism. Oh, it looks like I have a spelling error in there. I'm sorry about that. And so a better way to write this, nice job, is this. So here's our rewrite. Read that. See how that's different? This is a bacteria that's very virulent, known to cause apple blotch. Symptoms are caused by secretion of toxins. Some of them are largely responsible for disease, whereas others are less active and contribute minor toxicity. Very different language. It's repackaged and restructured, but it's not using this host-specific toxin, HST. I rewrote that to mean something a little bit more, use language a little bit more plain. And so this is how you would rewrite it for something like a scientific paper. And of course, you still have to cite it because you didn't create this information on your own. Do I have questions about this? This takes practice. It does. And I appreciate that some of you haven't gotten this practice yet, but now is your chance to practice it in this class. Okay. So primary literature. Most primary literature articles tend to follow a similar format. So this is abstract intro, methods, results, conclusions, sometimes also called discussion. Is there another word for that? Sometimes sometimes it's both. Actually, I've seen papers where they have a discussion and then they have conclusion, which is like sort of like a concluding paragraph. Some papers are a little bit different in that respect. And then they also have references, but they have each of these things. So if you're looking at a paper and you can't find a method section, you can't find a results section, you are not reading a primary research paper, you're reading a secondary source. You're reading a blog, you're reading a magazine article, you're reading a review paper. The order of these headers depends on the journal. So for example, in science papers, they might not explicitly show you the headers in a science paper, but it's written as more of a narrative. When I say science, I mean Science magazine. But you can, as you read through it, you can see, oh, it's actually got all of these parts, but it's just written in a more narrative format. Nature, I don't even know that they publish their methods up front. So I think it's intro, results, conclusion, and then they either have methods you can find online or like super tiny text where they talk about their methods. And so the organization of this might differ depending on your paper, depending on the journal, but all of these pieces will be there. All primary literature has these pieces. A lot of journals, especially if you're choosing something like Nature magazine or Science magazine, will have supplementary information that's accessible online. These are important details of the research, but are published online because of page limits and things like that in the journal itself. Because they are called supplementary information, a lot of people, even my colleagues, consider it to be like, oh, that's extra and I don't need to look at it. No, definitely you need to look at it. Take a look at that supplementary information. It might clarify things that are confusing to you. Or if you're reading through your paper and you think, really, I really would have liked to seen them try this. They might have actually tried it, but they give it to you in your supplementary information. Some journals, like I said, Nature and Science magazine, consider materials and methods to be completely supplementary. And so you might be looking for that when you're writing your paper summary and you can't find it. And it's because it's probably accessible via a link online. So make sure you access all of the information contained within your paper, including the supplementary information. Before you begin reading your paper, notice a couple of things. There's some information that's given to you up front. This is a review article that my colleagues at Mass General Hospital wrote in 2012 about cholera, simply titled Cholera. So it's just a review of current research, although it's not current anymore, research at the time that was current to epidemiology of cholera, treatments of cholera, where it is, who it affects, so on, things like that. Right up front, you'll notice that each author has these tiny numbers attached to them, and those are footnotes that are related to their institutions. So take note of their institutional affiliations. Things like universities and government agencies are well respected and sort of known to be institutions that engage in scientific peer-reviewed research. Some other might be research institutions, but are more agenda driven. And so you've got to be careful about where this information is coming from, because it could be written with a bit of a bias. All of you will be required to read peer-reviewed research. And so research that has been examined and vetted by other experts in the field that are not related to your research and not related to your goals specifically. And so they're impartial observers, but very educated observers, making sure that you're doing good science, that you've got all the information there, that you're drawing conclusions based on your results, and you're not drawing conclusions just because you want to, not based on any result that they can find. So am I reading a good journal? One way to know is to look at the author's affiliations. All of these authors are affiliated with either a hospital, Mass General Hospital, Harvard Medical School. This is the International Center for Diarrhea Disease Research in Dhaka, Bangladesh. So that's the equivalent to Mass General, basically there, and so on. And so you can see these authors have university or hospital affiliations. And so it's more likely to be less biased. When their writing will be likely less biased. You know, they're not trying to sell or promote a vaccine. They're trying to give you information. Also take note of the journal in which it's published. Are they reputable journals? Reputable journals are indexed by important databases like PubMed or Web of Science. Be very careful about predatory open access publishing. What this is are journals that say, we allow anybody to read our papers. Some of those journals are very reputable, and I love open access journals because you have access to those papers, right? You don't have to jump through firewalls and hoops and paywalls to try to get it. But some open access publishers are predatory, meaning they are driven by other motives and agendas. So make sure you're reading reputable journals. Anything you find in PubMed is probably fine. Anything you find in Web of Science, anything that we have subscriptions to are okay journals. They're all peer-reviewed, respected journals. If you're unsure, email me and I'll go through this process with you to show you like, oh, nope, this is not a good journal because, you know, it's all backed by pharmaceutical company or something like that. Not to say that good research doesn't come out of pharmaceutical companies, but you just have to read it with a different eye, you know, read it with a more critical eye, knowing that there might be some bias involved in the conclusions they're drawing. Impact factors is something you can use as a way to gauge how important, or not how important, but how reputable your journal is. So this website will bring you to, please, I hope this link still works. Oh, and I will post the slides after class so you have access to all this stuff too. We'll give you the impact factor for different types of journals. Okay, so I'm trying to find. So Nature Reviews, here's a good one. So Nature Reviews has impact factor that is very high. So they're using different metrics here to measure it, but here's basically your impact factor. So this one is up very, very high. And so that what that means is they are respected journal in the field. They publish cutting edge research, very recent research in you know, hot topic fields like CRISPR technology, gene editing. These are clearly the ones, those are the ones I read, or vaccine development and things like that. Sorry, those were all infectious disease, but that's how my brain works. That's what I study. And so you can see they're sort of ranked in highest to lowest order here. And so most journals are on this website. See where your journal ranks. See how good an impact factor it is. That's not, that's just to tell you like how many people are reading that journal and accessing that information. It doesn't necessarily tell you the science is good or bad, but it's one thing, one tool you can use to help you. Okay. All right, so the first thing you might notice when you open your paper is the abstract. Abstract is a summary of the entire paper, and I actually would recommend you read it last. Don't read it first. You might have read it, you know, usually if you're going to pick a paper. So during this week, when you're going through, say you've narrowed it down to 5 papers and you don't know which one to pick, probably what you were doing was reading the abstracts, right? Reading the summary of these papers. But when you, that's okay when you're trying to pick a paper. But when you sit down and it's time to digest that information on, you know, over the weekend, when it's time for you to sit down and really work through the paper, I actually recommend you don't read the abstract first, because you could end up biasing yourself towards how the author is interpreting the data, not how you yourself, the independent scientist, will then be interpreting the results and conclusions that are presented to you. So you can read the abstract, you can read it, but I recommend you don't read it first, because it might be better for you to kind of come at this with a fresh pair of eyes, is what I like to say, without that bias, like I am looking for this conclusion. You don't want to read a scientific journal like that, because you want to be able to read it critically, meaning, do I agree with what the author is about to tell me based on their methods and based on their results? Do I agree with the conclusions they've made? This kind of feels advanced, right? I don't know how many other classes have asked you to really dig into one particular paper like this, but this is something you do need to practice this skill and get used to. If you plan on going to graduate school or medical school, you will just be reading papers constantly all the time. And so if you want to make sure you come at it with a more of a critical eye, avoid reading the abstract first, kind of end on the abstract and see like, okay, I agree with what they said based on what I just read. The first point in that how do I read a paper, reviewing the paper summaries, or how to read primary literature, is make sure you can identify the big question. The big question is usually right up front. It tends to be in the first opening paragraph of your introduction, and it's setting the stage. What am I about to read? What topic am I going to dig into? And so the big question is not what the study is trying to answer. The big question is, this study fits in with all of these other studies, all trying to tackle this big question, this big problem. And so if you read this is the introduction to a paper that I wrote, God, I think in graduate school, it was a million years ago. So none of this is current research. It was so long ago, but this is the opening paragraph to one of the papers that I wrote. Take a minute and read that. Yeah, we do have this. If you feel comfortable in identifying the big question, please unmute your mic and I will call on you. I think there's like a raise hand. You can do that little icon, like something like that too. So take a minute and read this. See what you think. So I'm now looking at all your beautiful faces. Digest it, right? This might be the first time you've read primary literature this semester, probably. So read it. What do you think? What do you think the big question is here? What is the big problem that this paper is addressing with their small study, with my tiny, tiny little study? I'm contributing to a body of knowledge. And so this is really asking, what's the body of knowledge? What is the big topic? All right. Anthony, you have your mic on. Do you think you know what the big question is here?
Oh, I didn't know my mic was on.
I kind of figured that, but I picked on you, sorry.
All right, you're fine. I'm still looking at it.
Okay.
Actually.
That's okay. Anyone else? Bartan?
Yeah, I think it's how like physical environment. So temperature, for example, relates to increasing the likelihood of certain disease, like certain viruses, to have efficacy.
Yeah, so you're 99% correct, except Vibrio parahemolyticus is what a
Or a
bunch
bacteria.
of, like, it's a bacteria, yeah. That's how you say that, by the way, Vibrio parahemolyticus. That was quite the bug that I worked on in graduate school. And yes, Vartan, you're correct. The big question here is, how do fluctuating climates and temperatures affect pathogens that might live in the environment, such as Vibrio parahemolyticus? Do you get more food poisoning and bad toxin producing guys during certain climate changes or environmental changes? So that's the big question. Is that clear? Does that make sense to you guys? So, if I were reading this, if I got my paper in front of me, I've got my introduction, I would maybe write one line in my notes summarizing what the big question is here from this paragraph. I don't have to get into details about, TDH or TRH. Like, I don't really know what those are yet, and I'm sure I'm going to learn more as I read this paper. But for right now, the big question is, how does climate affect the presence of a pathogen, specifically Vibrio parahemolyticus? For many of you, your big question will be related to the topic you chose. And so some people chose something like treatments for cancer. And so your big question will be, if you picked a breast cancer paper, like what are the most recent advances in breast cancer treatments? That's your big question, or something similar to that. And that's going to be related to the topic that you chose. So it's bigger, it's very non-specific. Later in the paper, maybe, so this one tends to be, really depends. If it's hypothesis-driven research, so like they're trying to answer a specific question, usually this comes in the very last paragraph of the introduction, right before they start talking about methods or results. You'll usually get a hint at what specific question this paper is trying to address. So not, we're trying to address climate change and how that affects Vibrio parahemolyticus. No, that's too big. This one tiny paper is not going to address that big meaty topic. There are thousands of papers that address that topic. This paper is going to try to answer one very specific question about that topic. So take a look at this, read through this. See if you can identify the specific question this paper is trying to answer. Lots of technical language there, right? It's an evolution paper. My discipline was evolution and ecology of microbes. And boy, the language that people like to use in those papers, including myself, can be really technical and complicated. And so one thing you might notice as you're reading through this is, what the heck is multi-locus sequence analysis? I'll tell you, it's outdated technology. Nobody uses it anymore. But it was good when I was using it in graduate school. You might have to look that up. You might have to hit up Wikipedia, like, what the heck is this thing? Just to get a one-liner explanation of what that means. Or you might look up something like, maybe there's certain vocabulary in here you don't understand. What does endemic mean? What is recombination? I don't remember what that is. And so these are things that you might have to look up and remind yourself of as you're reading through these really technical papers. Can you dig out the specific question here? Anyone want to give it a try? So Vartan was brave. Let's see if anyone else wants to give it a try. I'm polite for like 20 seconds and then I start calling on people.
I'll take a guess.
All right, who's talking? That is Sharif. Hey, Sharif.
Hello. What affects? Oh no, wait. Oh my God, I had an answer in my head. What? How does the environment affect the strain, or like the DNA sequence of the strain?
Yeah, specifically that diversity. So how does something like temperature or salinity affect diversity? Yes, that is the specific question this paper is trying to address. And then using that information, we can get an idea of if these different, aspects of the environment, temperature, salinity, rainfall, affect diversity, does that increase or decrease the amount of pathogen? If we have more diversity, does that mean we also have more pathogens? And so that is a testable, specific question. We're going to go into the Great Bay. We're going to take samples out of the Great Bay. We're going to take environmental samples like salinity and temperature and see if we can correlate what's living in the Great Bay and what's happening in the environment. So that's your specific question. So this paper can address that. This paper cannot address how climate change affects the rise and fall of pathogens, but it can address a tiny, teeny, tiny bit of that. And so that's why it's the specific question, not the big question. Okay, so far we're only really digging into the intro, and this is not super technical, right? It's still like, okay, there's some words I don't understand, I might have to look up, but so far I haven't seen anything crazy technical until you get to the methods. And that's usually where people start to struggle. I don't understand what this method is. I don't understand what any of these words are. And so for identifying the approach, I highly recommend you do something like draw a flow chart, or sort of try to get a step, like try to step by step figure out what they did in their experiment. And so all of you right now, so this is a different paper than I was just previously talking about. This was one I wrote in, oops, hold on, my door is opening and I don't have a mask on, hold on. Okay, whew. All right. I'm sitting in my office by myself, so I feel like I don't have to wear my mask, but that door just started opening. I apologize. Okay, so this is a different paper I wrote as a postdoc talking about methods for, excuse me. This is a method, this came from the method section from one of my previous cholera papers. And so really where the approach is, so you see, this right here is the end of the introduction that I'm highlighting with my arrow. I hope you can see that. Urine and cell culture models have been useful for determining the role of these different things in the inflammatory response. However, there is a disparity between human and mouse inflammatory response. And so mouse models cannot fully replicate what we see in cholera patients. And so for this study, okay, so that is definitely going to, now we're starting to identify the approach. And in fact, this is still not in the materials and methods. This is really at the end of my introduction. And you will see that in some papers, they tell you the question they're going to answer, and they give you a quick, summary of how they approached the experiments that would address that question. And so see if you can figure out, there's a lot of language and, there's a lot going on here, but see if you can try to tease it out from this paragraph. What is the approach? So I gave you the end of that paragraph that comes above the for this study. to tell you what people were doing. And so for this study, we're going to do something different. We're not going to use a mouse model. We're going to do something different. What is it? What is that different thing? Normally I teach this class in 2.5 hours because it meets once a week. And so we have like lots of time to go through these. And so I apologize, a lot of the exercises that like I normally would do for this class, I forget. This is only an hour and 15 minutes. So, you know, again, anything that's in Top Hat that says we're going to do this exercise in class, simply attending class gets you credit for that. It doesn't really matter what we do. Simply attending class will get you credit for those exercises. So I got you, you're good. Okay, so does anybody identify the approach? What do you think? What's the approach that I took here to try to answer this question of what happens in the inflammatory response in cholera patients? How am I going to figure that out? I can't use mouse models because it's not exactly the same response between humans and mice. So what did I do instead? Allison, what do you think? Oh, Valeria, you've got an idea?
Yeah, you take samples from humans, like from patients with cholera,
Mm-hmm.
and then you identify if they have an innate immune response to it.
And is it clear from this paragraph how I know the difference between a response to cholera and a response to just being alive, being around? Like, how do I know it's specific to cholera, these responses that I'm going to try to look at?
I'm not sure.
Okay, that's okay. Does anyone think they know?
Is it because it says immune responses to V. cholerae? Like under when it says we then examine differentially regulated proteins and pathways to identify key innate immune responses to V. cholerae? Would it be because of that we know?
Yeah, you were so close, Brianna, so close. Differentially regulated proteins, like, So if you go to the line right before that, it says, we collected duodenal biopsy specimens. So again, technical language, if you've forgotten what a duodenum is, it's the upper tract, upper intestinal tract, about as upper as you can get, really. So we're going to collect duodenal biopsies from cholera patients during acute illness, and again, 28 days later during convalescence. And so you might have missed it because instead of saying, again, 28 later is when they're feeling better, I used a technical term, convalescence. And so what I'm doing here is I'm collecting biopsy samples from cholera patients while they have cholera. I have those patients come back 28 days later and I take another biopsy and compare. And so how do I know this immune response is in response to cholera? Because it will be up at day 2 when they have cholera, and it will be gone at day 28 when they don't have cholera anymore. And so that's the approach. I'm not going to use a model. I can't use mice. It's not really the best model. Cell culture is great, but it's so far removed from the person. I'm going to go right to the cholera patient, collect samples, see what's on, and see what's off later. And that will tell me what the innate immune response to cholera is in a person. I'm going to go to a person and collect samples. And so that's the approach. This is not, I'm sorry, I misspoke before when I said draw a diagram, draw a flow chart. This is not where you would draw the flow chart. This is where you would write like 1 or 2 sentences with the overall summary of what I did. I can't use mice. I went to the patient, collected patient samples, sequenced those proteins, and identified what was there. That's it. That's the approach. Okay, so here is where this is where it gets technical. Now I've just crossed from introduction into methods. I'm faced with this really terrible word right off the bat. Let's see if I can actually, endophagogastroduodenoscopy. No way, there's no way I can try that one. Professor Parvizi, does that roll off your tongue? It certainly doesn't roll off mine.
Yes, right. Esophagogastroduodenoscopy.
Yeah, exactly. What?
So it's a tough one. I agree.
It is a tough one, yeah. And it's certainly not obvious what that word means at all. And so right away, both me and Professor Parvizi would probably have to go like, what? Right. Like, what is this? What exactly is that procedure? And so too would you guys, right? You'd have to go look that up. Here's where it gets technical. This is all the technical stuff, technical information. And so it can be really hard to paraphrase this. This is usually where students hit the wall. Like, how am I supposed to repackage this into plain language? It's also technical. people. And it might help if you start with a picture. Try to do a step-by-step. Like first, we did some crazy procedure, right? We did this procedure, which is basically the process of taking the biopsy from the duodenum. So we went and actually, I think you go in through the esophagus, that's why it's esophago. So you go in through the mouth, get all the way down there, biopsy the upper intestinal tract, and we did this after clinical stabilization. So they came into the hospital, sick and dying, stabilize them for a day, the next day, take a biopsy.
Sounds like a very pleasant procedure.
Yeah, these patients are heroes. They really are. The people in
Exactly.
Bangladesh care so much about this problem that they will put themselves through whatever to try and solve this problem.
That's amazing.
Yeah, they're really wonderful people. They're just ravaged by this disease every single summer. It is not a pleasant procedure. And not only did we ask them to do it when they had cholera, but we then asked them to come back 30 days later and do it again. And they did. We had full compliance on this. It was unbelievable.
That's
They're
amazing.
so dedicated. Yeah. Sorry. This is my little plug for Bangladeshis who are like the best people ever. Okay, so we took some biopsies. We've got some technical information. We've got a weight range. We stored them. Then we did all these other. So what your exercise would be here is, can I figure out what the step-by-step process was from this paragraph? Is some of this information necessary? Is some of this information unnecessary? You know, so what am I going to write down? So probably what I would do is I would write down, okay, we performed the biopsy. I would leave out that word because it's like, that's not going to help you. So we performed biopsy on people on day 2. We took 5 to 6 specimens, all within a particular weight range. I don't have to get specific, right? You don't have to say in 40 to 120 mgs. No, they were all fairly even in weight. That is how you would translate that into plain language. Of these, so of the 5 to 6 specimens that were taken, 1 was snap frozen in liquid nitrogen, and then others were preserved on microscope slides. I don't have to say -80. I don't have to say paraffin preserved. Nope. Some were frozen. Some were preserved on microscope slides. Here it says endoscopy was repeated 28 days later following presentation, and a similar set of samples was obtained. Did it again a month later. Due to destructive sampling of the biopsy specimens used for proteomic analysis. What? That's, again, so much technical language. So how do you break that down? You might look up the phrase destructive sampling. What does that mean? You might look up proteomic analysis. What does that mean? I can retranslate that phrase into, because when you sequence a sample, you cannot use that sample a second time. So we use some samples for sequencing, and then different samples collected from the same person for other types of experiments like cytokine measurement or lymphocyte extraction. And so we can't just take one protein sample and subject it to multiple different experiments. We're doing lots of different experiments, but we're doing it with the different samples taken from a single patient. And so it might take you a minute to read through that a couple times, write it out, like what's happening with this one? They said a similar set, what's happening? Draw arrows, draw pictures, but leave out the details like -80 degrees Celsius, 40 to 120 milligrams. That's not necessary in a summary, and it's not necessary for you to report it, because this is just the author's way of saying we did good science, we were consistent in our collection, and I'm telling you exactly how we handled everything. But you don't need to tell us that, especially since when you go to present this information as an oral presentation, you are given 10 minutes and that's it.
Professor Ellis, I don't know if this might be helpful to them, but the way I would have it is to think that, you know, you're trying to make this comprehensible to maybe a middle school classroom. Seriously. You know, this is, and that's kind of ironic because this is like your capstone. It's an advanced biology course, but that's the whole point of it, communication. You know, your goal is to take complicated information and make it comprehensible to people who are not as scientifically fluent.
Yeah, it's a really good point, Dr. Parvizi. I think it will, we're going to talk a lot about that next week too, when we talk about knowing your audience.
Right.
Who is your audience? Do you know who's sitting in your audience? I won't get into the details, but I've given presentations before, like all that Vibrio parahemolyticus stuff. I had to give that presentation to a room full of fishermen so that they understood the risk they pose going out and oystering during certain high weather events, like high rain events. I had to talk to a room full of fishermen about multi-locus sequence typing. That was hard. That was really tricky. But also in the same vein, I have given cholera presentations at conferences in sort of a room full of cholera biologists. So I'm not going to waste my time in that room talking about, well, cholera causes gastrointestinal. Yeah, everybody knows that. They're all cholera biologists. So I'm not going to waste my time talking about that. So knowing your audience is really important. Right now, as you're working through your summer, your audience is you. And so Professor Parvizi's advice is really good. If you can repackage this in plain language, something that you could tell a friend, then that also is going to help you understand it too, because that's what you're used to reading. You're used to reading and interpreting plain language. Okay, I think we've talked about this enough. Again, here, obtaining human subject approval for this study, you don't have to tell us that. It's sort of assumed if you're standing up and giving a scientific presentation that you did this as ethically as possible and you didn't break any laws. And so like, you don't have to tell me that in your summary that you didn't break any laws. It's sort of assumed. Okay. Moving away from the methods, and now we're into results. This is from that same paper where I'm talking about, I took samples from cholera patients from their upper intestinal tract, and then I sequenced proteins found in those samples. So what proteins are present and in what amounts are they present was what I was able to answer with this. And so here's my first results. And again, oops, sorry. It's getting technical again, lots of technical information. A total of 4,612 proteins were identified in 8 paired acute and convalescent phase samples based on protein spectral count. Already I got to stand back. What does protein spectral count mean? What does convalescent versus acute? What does paired mean? And so make sure that you understand this language so that you can communicate it in plain language. This will take, again, you got to read it multiple times, take notes, look up things. You cannot simply just read this paper and be like, yeah, I got it, let's go. Nope, you're gonna have to read it a bunch of times. So I want someone to try this. This is technical, so it will be tricky, but can somebody try to read this? What is this figure saying? What is this figure telling me? The text will help you because anytime there's a figure presented in a scientific document, it's referenced someplace within that document. And so this is figure 1. What is figure 1? Oh, it's a heat map, representation of proteins showing, okay. And so I'm going to look at this figure. What is this figure telling me? What does the red mean? What does the blue mean? What is all the tiny text along that column? What are the sample names? Like, what is this? What does this mean? Take a minute. So the first step in looking at results is, what are the results? Not what does this tell me about the question, but what is this figure saying? What is this figure showing me? Don't interpret, don't conclude, right? We're not concluding here. What does this figure show me? And I'll tell you right now, I got a lot of heat from reviewers about this figure. But I won in the end because it's published now. So what do you think? Anyone brave enough to try and tell me, what does this thing mean? Some of you have picked very technical papers that have things like box plots and probably data that you're not used to interpreting and looking at. You can, of course, reach out to me to help you interpret this, but you're going to get technical stuff like this. So what is this telling me? What is this figure showing me?
Is it showing you for the, it's a heat map representation of proteins. So this kind of goes back to, I think, what you were saying with during the infection process and then the 28 days after, so post-infection. So I think this is showing you that So the heat map would be like the darker would be, or the red, I should say, of the proteins during infection, and then 28 days post. So you can basically compare what proteins and therefore what pathways are activated as an innate immune response during infection, and then how that changes 28 days after.
Yeah, nice job, Sarah. That was very good. So yes, the intensity of the color tells you how much of the protein is there. And so very intense red means there was lots of protein. So for example, this is one of the very top says LTF. You probably can't read that because it's super small. That's one of the criticisms I got from this picture. But LTF, so that seems to be very red in most patients, except for patient 5 and 6. And so these sample here are telling you patients 1 through 8. In patients 1 through 4, LTF was really expressed during disease and not so much during convalescence. It is super red because it was really on when they had cholera and off when they did not have cholera. For this one in the center, there was no change in that protein in these 2 patients. And so what I can conclude from at least that one protein, so these are protein names on the side, is that that LTF protein may be important in cholera because it seems to be expressed in most of these patients, but not all of these patients. This figure is so hard to interpret, and really, if I were to draw one conclusion from this figure, it's that no 2 people have the same response to cholera. And that's probably all you can really say based on this paper, or based, not on the paper, based on this figure. There's more you can say with the paper. So yeah, I've looked at all these proteins. Some are really on during cholera, some are off, some of them didn't change at all. Some of them are really highly expressed when they're well and not at all on when they're sick. But what we can definitively say, we can't say any one protein is important, but you can certainly say everybody had a different response to cholera. And that in and of itself is information. That kind of tells you, well, we should probably be looking at more people rather than 8 cholera patients. It's not really enough to say anything. But it's certainly enough to say that this is a complicated problem. People have very differing immune responses, innate immune responses to cholera. Professor Parvizi, did you want to say something?
Yeah, it says something about sample size, right?
Certainly does. Yeah, when I wrote this paper, this is what's called discovery-based research, meaning I wasn't trying to say anything too specific with this. I'm just sort of seeing what's there. Like, is this a good jumping off point? And it turned out that it was. I won't get into the details. People interested in cholera, you can ask me later. But this was a really good starting off point for me to really narrow down what was important in cholera. But again, 8 people, this basically tells us not really enough to say too much other than that everybody behaves differently. So that's basically it.
As a visual artist person, though, I love this chart.
Oh, thank you.
Yeah, from that perspective. Yeah, it's actually very informative, you know, the intensity of the colors.
Mm-hmm. And you guys will see a lot of that variety in your papers. You'll see these images. It's not simply, you know, line graphs anymore. People get really clever. There are Photoshop geniuses out there that I envy. I don't know how they do it, honestly. Okay, so we're closing in on 10:45, so I'm just going to keep moving and hopefully we can just get through this exercise. Okay, so the other question you want to be asking yourself is, do the results answer the question? If I look at this figure, does this somehow address the question that was being asked at the beginning? This is a different paper. So this is experimental evolution of bacteria to study the extent to which adaptation can affect host range. And so these ones, if you read through these figure legends, and figure legends are so important. You like every time you're looking at a figure, start with that figure legend. It's going to tell you what the squares mean, what the open symbols, what the closed symbols mean, what the lines mean, all of that, what the axes mean, how it was measured. You got to read those figure legends. Do not ignore them. Okay, so here we have adaptation by 12 different populations. So it looks like different lines represent different populations. These are generations, so how long they were evolved. And they were evolved in this onion medium, this particular environment. And then I'm using technical language here. Fitness was quantified by direct competition with the ancestor. So fitness might be something you have to look up. Fitness in this example simply means reproductive success, doubling of bacteria. And so we've got 0 generations where they're not evolved. And then we evolved them in onion media for 1,000 generations. And then it says that we are quantifying fitness by comparison to the ancestor. So the ancestor competed with itself would be 1. anything that shows an increase in adaptation is above 1. And so if I'm looking at this figure, I see, well, I've got 12 populations that all have higher fitness than the ancestor. Okay, so they seem to have adapted to this media. Then when you look at this other figure, we have nematode mortality, death of nematodes over time. These same symbols, so these are the same populations. And now if you look at the figure legend, we see that this is nematode mortality as a consequence of adaptation to a different environment. And so do these questions, or do these figures answer the question, did these populations adapt? And do they now have difficulty surviving in other environments? So did they adapt? Well, if we look at our figure, we can see that our numbers are above 1, our error bars don't overlap. And so yes, they adapted. They seem to adapt to onion media. They got better than their ancestor. They reproduced more in that environment than their ancestors did. When you look over here at the other figure, we see, if you look for that wild type, so that's the ancestor, and you can see that WT is the ancestor because it's given to you in the figure legend what that means. And so it looks like the ancestor starts killing nematodes around 70 hours and above infection, whereas all of the other populations, with the exception of maybe one, weren't able to achieve the same mortality that the ancestor was. And so does adaptation to one host restrict adaptation in other hosts? These figures support that conclusion. We adapted in one, we adapted to the onion media, they're better than their ancestor, but it looks like they can't kill nematodes anymore. And so they're struggling in that nematode environment compared to the ancestor. And so this is what you should be doing when you're looking through your figures. What is this? What am I looking at? What are the measurements? Can I write one line that describes what I'm looking at? And then ask yourself the secondary question, before you go into the conclusions, before you even do that, does this figure actually address some part of what they're trying to prove? That is a really critical piece. And this is something I really wish that I could, like if I were giving the 2.5-hour class, the next exercise is to read 2 scientific papers, which you should have done before coming to class, but I'm betting many of you did it. So that's why I'm not doing that exercise, because I forgot to remind you, it was embedded within Top Hat, but I felt like this was a better use of our time, is going through systematically each of these different points. But anyway, normally what I would do is you read these 2 scientific papers, and one of them is based on false data. So one of them is a tried and true paper, peer reviewed, supported, all the way. The other paper is bad science. And so in class, what we do is we try to figure out, why is it bad science? This is still something you can do on your own because you have access to the papers. Why is this bad science? And one of the reasons that we come to as a group is that the figures being presented in one of those papers does not address the question they're trying to ask. It doesn't really answer the question they're asking. It might answer some other question, but not the one that they're asking. And so it's bad science. And so that's what you should do with all of your figures. Get them all in one place, look at them, your one-liner about what this figure shows, what it measured. Does this figure actually contribute to answering the question they asked at the beginning before you read the conclusions? Because then once you get into the conclusion section, the authors are going to try to convince you that what they did supports their conclusion. And you need to be convinced, but you need to have some idea of what you think is the truth before you start reading that conclusion, so that you can critically evaluate these papers. Does the information actually answer the question being asked? Most of the time, it's going to be yes, but some bad science gets through the door and does get published. The bad scientific paper that I would normally have you guys critiquing was published in Science magazine. And so it was missed, but it's bad science. If you read through it, you can see the figures do not support the conclusions that they're drawing. And that's ultimately how you read a paper with a critical eye. Does the experiment, do the results support the conclusion actually being presented to you? That's also what reviewers do when they're peer reviewing research. Okay, I don't know how many slides I have left. I think that was, do the authors interpret these results? Yes, so basically everything that I just said to you. So that being said, I will post these slides. So if you want to try these exercises again and go through the recording and try and practice the skill, I will post the slides and post the recording so you can practice again. Do you guys have any questions before I end class? I know I'm 5 minutes over, so I don't want to keep you. I know you have other courses to get to.
You guys, just a couple of questions I have as far as attendance. Maria Miguez Ramirez, are you here? Maria?
Yeah, I go by Sophia.
You go by Sophia. Oh, okay. That's a little confusing, but I'm going to try to correct that for next time. Thanks.
Sorry about that.
That's all right.
Sophia,
What
I've met you
about,
multiple times too, so I remember.
uh-huh.
Yeah.
What about Inderpreet Thind?
Oh,
Inderpreet? Okay, so
Yeah, that's
Inderpreet is the only person missing here. Everybody else came in, which is awesome.
Excellent.
Great.
I'll reach out to her and make sure she's okay.
Okay.
All right. Any questions about what's expected of you? If you have trouble finding a paper, don't email me on Friday night and say you have trouble finding a paper. No way, do that on Thursday night so I actually have a chance to help you. Get that paper figured out for Friday so that you can start doing this process of summarizing and interpreting your paper. I highly recommend you don't pick a paper that has 18 figures. Don't do that to yourself. That's going to get hard. You're going to spend a lot of time ripping through that paper, and you'll probably have a really hard time trying to figure out what to present, because in science, you were only ever given 10 minutes to talk. It doesn't matter how much you want to say. Too bad, you're given 10 minutes and that's it. Any conference presentation that I've ever done has been 10 minutes long. So this is something that we got to get used to in this class. It starts with the summary. Questions, concerns?
I just have one quick question.
Go ahead, Sarah.
Sorry. When you say submit our notes to Blackboard for credit, the notes that we took in the videos, are you going to be posting a link specifically?
Yeah, I gotta put up the assignment link for that. So I'll do that right now.
Awesome, thank you.
You're welcome, Sarah. Yeah, I think oh, sorry, go ahead.
I'm sorry, Professor Ellis.
Go ahead, Najee.
So is what she just asked about, is it the notes that we took today for today?
No, it's the notes. So if you go to Top Hat Chapter 1, Exercise 1, you have to watch a video and take notes and then submit your notes to Blackboard. It's just the exercise in note taking.
Okay, gotcha. So we're never gonna be, are we ever gonna be submitting what we do in class or no?
Oh, no, because I take attendance and that's it.
Gotcha, okay. Thank you.
Oh, you're asking that because you were in 2 of my courses last fall.
Yeah, I was waiting for it.
Yeah, nope. Simply attending class gets you the credit. I have to do it that way for those other courses because attendance not required, but it is required for this class. Yeah.
Gotcha. Thank you.
You're welcome. All right, guys. Did something happen in the chat? Nope. All right.
Professor, I have a question.
Yes, go ahead,
so
Barton.
for Top Hat, exercises 4 and 5, we said we were going to discuss like in class and we didn't get to that. So for this like chapter 1 to have it complete, we just have to answer like all the other questions and
exercises 1 through 3, 1, 2, and 3. For, like I said, anything that I say I'm going to do in class, simply attending class gets you that credit.
Okay.
Yeah. That's, again, if I had 2.5 hours, we would have done all those exercises, but I kind of got to repackage things for online learning. And I didn't want to completely rewrite my textbook for 1 semester. So definitely ask questions if you are confused about things, but there'll be stuff that's like, we'll do that in class, just attend class and you get credit for that.
Okay. Just avoid my email that I sent you a couple days ago or yesterday to the.
Sure, no problem.
Okay. Thank you.
You're welcome. All right, guys. Thanks for staying a little late to answer some questions. I will see you next week. Reach out to me as much as you need if you need help. Have a good rest of the week and be well. Stay safe.